Yes, that was a good example of what's claimed to be evolution, and it's certainly changed. But notice how often they referred to gene loss. So mutations certainly are real. They happen, they do change things, but they don't make anything evolve. So let's have a look at that. A mutation is simply a change in genetic information, and they are considered to be what drives evolution. This is a quote I found in a scientific paper a few years ago, but this is basically the mindset that the evolutionists have. Mutation is the engine that drives evolution and adaptation forward, in that it generates the variation on which natural selection acts. Now, that's an important. If you remember back to a few weeks ago when we did a couple of programs on why evolution won't work, and we concentrated on natural selection and made the point that selection just literally selects things. It chooses things from already existing options, but it doesn't explain where the original options came from and where the changes came from. It's just a means of allowing some things to survive and other things to die out. So if you combine mutations and selection, the standard sort of story is that mutations result in good and bad changes to genetic information. Natural selection weeds out the bad mutations, leaves the good ones, and so over time the good ones should build up and produce some kind of new living thing. So overall, somehow we started out life as chemicals, and then by a whole series of processes, including mutations, over a long time with the struggle for life and the war of nature, somehow we came into existence. So we've gone from molecules to human beings. And well, that's a nice story, but there is a problem. There aren't any good mutations. There aren't any mutations that actually improve structures and functions. Now, did you notice in Craig's presentation about seahorses and pipefish, the number of times they referred to gene loss, genuine change, but not improvement and not new structures or functions? Now, none of the mutations we have observed build whole new structures and functions. And this is no surprise when you consider how mutations come about. So, what are the things that cause mutations? Well, basically, there are external and internal things, chemicals and radiation from the outside. And then there are also copying errors. In our cells, we are constantly copying DNA, and sometimes that goes wrong. We also have repair mechanisms, so there are lots of backups inside our cells, but sometimes even the repair processes don't work or get knocked out. So it's interesting that if we go back to the external ones particularly, whenever there's been an accident and there's been a massive sort of pollution or a nuclear problem, like with the nuclear reactors in Japan and Russia, do you hear any evolutionists come up and say, oh, well, just there'd be lots of wonderful new things that are going to evolve? No, the environmentalists, who are all evolutionists, will wail and moan loudly that we've messed up the environment. And yes, we have, but nobody's claiming that anything new is going to evolve out of that. In fact, we want to stop mutations. So the results of mutations are that genetic information is degraded. Chance random processes, which is what you get from those external and internal problems, always result in gene loss. So randomly knocking around the genome does not improve things any more than if you randomly knock around some computer code. That's not going to build a better electronic devices. Now, occasionally you can get neutral changes that don't cause disease or disability, but random processes don't add new genetic information. what we see is gene loss with the examples that Craig gave, and those are not unique. So let's have a look at maybe this applied to supposed human evolution. Now, back in 1999, I came across this article called The Odds of Losing at Genetic Roulette. Now, like gambling in a casino, chance random processes, eventually you lose, or very quickly you lose. And in this article, they made the point the number of harmful mutations that arise has been measured, and it is surprisingly high. Now, this was just before the first draft of the human genome, but there had been a lot of studies of individual genes and groups of genes. And so these scientists who wrote this review article had gathered together a lot of this information, and they came up with the conclusion that with each generation, there were 3 new deleterious, that's damaging mutations per person per generation. What's the significance of this? Well, keep in mind that they believe that human beings evolved from ape-like creatures over at least 3 million years, possibly as long as 6. And they went on to make the point that every deleterious mutation must eventually be eliminated from the population by a premature death or reduced relative reproductive success. Okay, so that's natural selection, right? Survival of the fittest, struggle for life at work. And what happens with the damaging mutations is that the creature that is carrying them will eventually die. So you have a genetic death. Now, if you add all of that up over supposed millions of years, you end up with 3 genetic deaths per person, if we believe that. And they asked the question, well, why aren't we extinct? If all this gene loss is going on, what's happening? Now, since then, we have had a real revolution, a complete revolution in genome studies. We've had the original Human Genome Project, and then we've had a whole heap of other genome studies. And we're learning a lot about how genes work. And one of the things that's important, it's not just the genes for our body structure and our enzymes and how they work, but also how genes are regulated. And so we're getting in some really interesting results from various projects that have come out of the Human Genome Project. This was an interesting one. It was called the 1,000 Genomes Project, but in fact, they ended up analyzing far more than just 1,000. It started out looking at 1,000, and back in the early 2000s, that was considered to be a fairly ambitious project. It still is. And they looked at human genomes from various ethnic groups. And one of the significant things they did was they managed to get a few groups of genomes from succeeding generations so they could see what changes were happening as you move from generation to generation. And here is some of the results that have been reported. There are lots of reports that arose out of this project and other similar ones. Now, notice this. On average, each person is found to carry approximately 250 to 300 loss of function variants in annotated genes, and 50 to 100 variants which are previously implicated in inherited disorders. We've known about genetic diseases for quite a long time, and we're gradually drilling down to find out sort of what exactly has gone wrong, with the hope that maybe we can do something about it. Here's another interesting result that came out of some of these genome studies. This was a headline in a general news service called news.com. Human beings are getting dumber. Now, we think we're at the top of the evolutionary tree. We're getting smarter. But this was from a review article called Our Fragile Intellect from Trends in Genetics. And recent human genome studies reveal that there are, per generation, about 60 new mutations per genome and about 100 heterozygous mutations per genome that are predicted to produce loss of function. some of which are likely to affect genes involved in the human intellect. Right, so we're talking about lots of gene loss, or at least loss of function, in that genes are becoming less effective, even if they are not completely lost. Now, there are all sorts of estimates flinging around, so you will see different numbers. And in general, the overall results from looking at all of these genome studies is, well, there are defunct genes for all of us. So we should ask the question, well, why is anyone alive and functioning if all of these genes are going wrong at such a rapid rate? Part of the solution is that genes actually come in pairs. And normally, if you have one defunct gene or one down-regulated gene where its function has been diminished, right, that is compensated by a normal gene. So we all carry 2 copies of just about all of our genes. And when these are passed on to the next generation, only one of those copies will be passed on to the next generation. So let's have a look at that process. So if you have 2 parents, one of whom has a mix of normal and defunct genes, and the other has normal genes, with each generation, the pairs get split up and only one gene from each parent gets passed on to the offspring. So you have mixing and matching. So the possible offspring from these 2 parents would be 2 offspring that inherit 2 normal genes and 2 that inherit a mix, normal and lost ones. Now, supposing you get 2 parents who are outwardly normal, have normal genes, or a normal function from that gene, but are carrying the defunct one. When they get separated and remixed for the next generation, there is a 1 in 4 chance that you end up with the 2 defunct ones coming together, and effectively that gene is lost. So that's the genetic death for that gene. Now, if you then go to a bigger population where you've got a whole lot of people, some of which will be silently carrying the defunct genes, after generations of mixing and matching within that population, you will get the 2 defunct genes coming together, and that gene is lost for this person. Now, this is a serious problem in small groups, in small populations that can only interbreed with one another. And in fact, this is the reason for the ban on close family marriages in most cultures. Even without knowing any genetics or about DNA, people have observed that when you've got a small population that only breeds within one another, you do get genetic diseases, and some of those result from genes just being lost. And so in our normal population, after however many generations we've had since the beginning of human beings, we're all carrying around inactive genes. Now, this was an estimate that was made quite a while ago. It's probably changed now, but in general, we are all carrying around inactive genes. So sure enough, and this was another project that came out of that 1,000 Genome Project. And so every newborn's DNA carries more than 60 new mutations, some of which lead to birth defects, including cancers. We have to remember that cancer is a disease where genes have gone wrong during our own life, but some of us actually carry a propensity for getting cancer. So that's defects that we've inherited from the previous generations. So there are new mutations for all of us. Mutations definitely are us, but we're not evolving. We may not be dead, but we are deficient. Okay, even if we go from that estimate of that particular project I just referred to then, right, 60 new mutations and 20 missing genes. Now, this fits with the biblical history of the world rather than the evolutionary history of the world. If we go back, look at the big picture of the history of the world, it started out very good. We see evidence of design, of complex creatures, like the seahorses that Craig showed us. But the world didn't stay that way. It's been affected by human sin and God's judgment. And since then, a whole lot of bad processes have gone on, including mutations. So, yes, there's lots of change, but eventually they lead to loss and death. Now, sometimes the evolutionists will say, well, some mutations survive for generations, and they become fixed in the population. Does that make them good? Well, here are just briefly a couple of other examples of how mutations may become fixed in a population and survive. Anyone recognize these? These are wild budgerigars. Wild budgerigars live in Australia. They're actually a type of small parrot, actually, and you get flocks of thousands of them, and they are all uniformly green and yellow. They're very, very, very pretty little birds. And if you look at them closely, there's, yes, a bit of variation in their body color, but basically they are green and yellow. But if you have budgerigars as a pet, you'll notice there's quite a bit of variation in their color, and you get budgerigars that are blue and white, and those are quite popular. So what's happened? Have the budgerigars evolved some blue genes? Well, no. In fact, it's actually a gene loss. The white head comes from an absence of the yellow pigment. Blue is actually a structural color in birds. Now, what that means is it's not a blue pigment. It's actually a microscopic structure. If you look under a microscope at the structure of the feathers, they have these very, very fine, it's nanotechnology. God invented that, we didn't. And where light is scattered and reflected, and that's how you get the blue color. So to get green, you have a combination of green and yellow. And so if there is no yellow pigment, the blue structural color is what you see. And people like that. And so they have bred these deliberately. And so you have got selection, it's artificial selection, and you do maintain this. So that's one type of selection where you do get a gene loss becoming fixed in the population. Now, the evolutionists might concede on that one and say, all right, that was a deliberate selection, but they still believe that mutations and selection work out in the wild world. Are there any examples of that? Well, here's one that we did report about in our newsletter only fairly recently in 22 rather. And we answered the question, can mutations produce a major change in body form? So just fiddling around on the outside, can you get a major change in body form? Well, we've seen one example of that with the seahorses and the pipefish. You did get, quite a significant change in the body form, but as Craig pointed out, it's actually a loss. And can natural selection preserve these sorts of mutations? Well, this was an interesting example in a plant. Now, anyone recognize these? These are very popular garden plants called aqualegias, or sometimes called granny's bonnets. And we reported about these because there was an interesting claim made about a mutated version of these, which were called hopeful monsters. Now, you may not know that term. It's rather a quirky summary of a theory that came out in the 1930s from an evolutionist named Goldschmidt. It was revived in the 1970s by Stephen Jay Gould and some of his colleagues, that A mutation of a single gene can produce sudden large changes, and so that was where the term hopeful monster came. The word monster does not originally mean big things. It means things that are changed in their form. The word itself has changed its meaning. Maybe that's a type of evolution, but in fact it's creation, because it has to go through a human mind in order to happen. But anyway, back to our plants. This theory disappeared back in the 1970s, basically from lack of evidence, until there was this recent claim where some scientists were studying a particular type of flower, variations of Aquilegias, and they found a mutated flower that looked quite different, but was the same species or the same kind as the other Aquilegias, and it's called um, a hopeful monster because it is significantly different in its flower form, and yet it grows out in the wild and survives. It hasn't been deliberately bred just for the florist trade. And what they found is that evolution can occur this is the claim that they made, evolution can occur in a big jump if the right kind of gene is involved when it's broken. Now, notice again, you've got this evolution by gene loss or evolution by gene breaking, right? When it's broken, these instructions aren't there anymore, and that causes a completely different organ, a sepal, to develop. We did not have a good example of a hopeful monster due to a single change until now. So they've made quite a big claim, right? We've found this hopeful monster. And they deconstructed, very helpfully, they deconstructed the flower to show us how this happens. So on the left, we have the wild type flower, on the right, the mutant flower. And if you pull it apart, you can see the components that make up a flower. It has sepals around the outside, then a row of petals, and then the reproductive parts, which is the pollen and the female pollen receptive part. Now, notice the main difference, or the only difference really here, is that the mutant flower has 2 rows of sepals. Now, note that the wild type flower already had sepals, so nothing new has developed. So they haven't generated anything new. And the interesting thing is, why did this plant survive when it's actually lost a whole structure? And that is significant. Notice that the petals have these sort of long tubes projecting from them, and that is where the nectar is. Now, normally these plants are pollinated by moths, which have a long proboscis, which goes down that tube where the moth can drink some nectar. But in order to do that, it needs to bring its head close to where the pollen is, and so that's how the plant transfers its pollen. How did the mutant plants survive? There must be something good about them. Well, yes, it's genuine survival. Aquilegias can be pollinated by bees. They produce plenty of pollen, and the bees will collect and transfer pollen, and the mutated plants still have pollen. Now, the other interesting thing is that deer and aphids normally eat these plants, but for some reason or other, the deer in this local area and didn't particularly like them, and neither did the aphids. So they survived. So we have real survival of the fittest here, but we still don't have any examples. So this is a rare example where one gene has actually changed the body form of an organism, but let's be a bit more generous and give the evolutionists some wriggle room and let them have multiple mutations. Can you get a complete change in body form with multiple mutations? Well, in fact, you can, and it's interesting that evolutionists claim that 4-legged, short-bodied reptiles like a lizard evolved into legless, long-bodied creatures, which we call snakes, and they believe that that happened by evolutionists. But then they turn around and scoff at us if we claim that snakes used to have legs and God somehow removed them. There's a bit of hypocrisy there, but we do need to ask the question, how did snakes get legless? Because genetics has enabled us to understand how that process happened. And the interesting thing is there are a lot of genes involved in this, but there's one particular one that's very prominent, and it rejoices in the name of Sonic Hedgehog. And honest, that is its name. And Sam, you would be interested to know it was named after a video game.